Tonight is Science News of the Week, and we're chatting about the cool science news of this past week. And this one that we're on right now is quite wild. It is on drug-resistant bacteria and how to maybe utilize drug resistance in bacteria and actually try to fix it. This is a super, super cool paper, one that is mind-boggling to me, and it makes sense, and yet at the same time, I'm surprised it hasn't been tested before. So, bacterial resistance, right? That's a thing. That's a problem that we're dealing with today. Bacteria are resistant to all kinds of antifungals, antibacterial, antifungal. We have fungal resistance. Bacteria are resistant to different kinds of soaps and other things that we're trying to use to get rid of them. Is there an existing mechanism that we can make them be less resilient that way? The reason I thought this was a really, really cool paper is because it makes no sense as to why we didn't think of this before, right? And that is probably the coolest kind of science. And this one that we're on right now is quite wild. It is on drug-resistant bacteria and how to maybe utilize drug resistance in bacteria and actually try to fix it, in the sense that it would be a way to fight drug resistance in bacteria. And it's by stealing and hijacking the natural machinery that exists in bacteria. So we're going to go over it in a little bit more detail, but bacteria are able to acquire genetic information from other bacteria in a process called conjugation. So you can give what is called a plasmid to one bacteria, a small circular piece of DNA, and then the bacteria is like, I want to share this plasmid with my friend over there. And so it sends out a small tube. Through that tube goes the plasmid. That's this one piece of DNA from one bacteria, it goes into another. And normally bacteria chop up viral DNA or DNA that's not theirs. But the plasmids, they're like, oh, hey, thank you. This is great. This is delicious. Let me incorporate you. And we were trying to figure out in this study, how does that happen? Why does that happen? That seems so fundamentally surprising. Why is it okay for a bacteria to accept a unique chunk of DNA from another bacteria, but not from a virus? Why is that happening? And the researchers have identified it. So this is a paper published in Nature called Diverse Anti-Defense Systems Are Encoded in the Leading Region of Plasmids. To break it down for you really quickly, a plasmid, again, is a circular piece of DNA. It becomes, instead of a circle, a line, a straight line, as it goes from one bacteria to another. And the beginning sequences, the starting genes at the very beginning of that formerly circular piece of DNA, are protective genes that protect the strand from being broken down. And then it reforms into a nice little circle, and then it's utilized as part of the DNA. And researchers want to identify how we can use this to treat bacteria that have become drug resistant. So basically creating drugs that now will have this region at the beginning, this little protective capsule, so hijacking the antibiotic resistance that a bacteria has through this plasmid and having it be treated against it. Does that make sense? We'll go into some of the more of the details of it too, but it's a really, really exciting bit of data to have there. So again, it's a way to counter antibiotic resistance in bacteria, and so is that unique genetic material that goes from one bacterium to another, and we would just be using the exact same kind of methodology. So before we get into, again, the details of some of these terms, I just want to highlight how this study started. There was a student in the lab, and he looked at 33,000 of these plasmids, those small circular pieces of DNA, 33,000 of them that were identified in labs that were naturally occurring plasmids from different bacteria identified in lab settings. And they wanted to be like, all right, of these 33,000, so yeah, corn, 33,000 sequences this guy looked at, and he was trying to find commonalities between 33,000 small circular pieces of DNA in bacteria. And he did find a group of genes that were always at the beginning of the plasmid when it turned into a linear plasmid, a straight line instead of a circle, straight line. And they're like, maybe these are anti-defense genes. Basically, the idea being is that these genes concentrated at the very beginning of the linear segment. So like when the circle was cut and be into a straight segment, all of these genes were at that cut site. So they would be at the tips of a single strand of this DNA. Maybe there's something to this. Maybe those are protective. Maybe they're doing something. And so of the 33,000 genes, he identified a subgroup, and it's like, maybe these are resistant genes. I'm not sure if so what Saphira's mama, he luckily didn't go through it manually at first. First, he wrote a computer algorithm to find commonalities between this data set, right? So the researcher needed to find what is the data that is common between these genes. And so you're taking 33,000 specimens, and the idea is there's something that is going to be similar between these specimens. And so what he found is the types of genes that are at the cut site that makes you go from a circle to a straight line. Everything at that cut site, there was some conservation there in the terms of the types of genes that were visible. The reason that's really cool is because, okay, there's something special there. Let's try to figure out what is that special something that's located at that cut site. And so the way to demonstrate that that cut site is what's important, there's actually a really cool way of experiment of doing this. And actually, I'm going to draw this out, y'all. I know we don't usually draw stuff out all the time, but Corn Relish was like, Balint, I expect you to draw this experiment. And so Corn Relish, for you, I will draw out this experiment, madame. I will show you that we can do this and we can draw out exactly what happens. The idea is a plasmid. All right, so this is a plasmid. It's a circular piece of DNA and it's inside of a bacterial cell. And then here is bacterial cell number 2. That circular piece of bacteria needs to go from this location over to the neighboring cell. It is to scale insofar as that circle cannot fit through the tube as a circle. It needs to be cut so that it can be a straight red line that can pass through and can then go back to being a circle. Are you with me, Spartacus? So it doesn't cut itself, Corn. There's machinery inside of the cell that will do the cutting for it. So what will be a little bit more to scale here is that is the DNA that's already in the bacteria. A plasmid is a really small circular piece of DNA. There's already a big circular piece of DNA in the bacterial cell. And that will have the code to cut something that's circular to make it single strand. And then it refuses as well. So what's cool about a bacteria, if it takes in a plasmid, it will use that as a template of genetic information. And it will use that genetic information to make proteins and direct behaviors of the cell. And so that's actually a common thing that we do in the lab is that we transform bacteria, meaning that we get a bacteria to take up a plasmid that normally is not in it, and that'll turn on and express genes that normally it hasn't before. So it's a way of acquiring it to have certain new features and traits of potential interest. I hope that makes sense. But so that's why these sizes are different to it. Okay, so the hypothesis, Sparky, we're gonna make a brand new one here. The hypothesis is that when you cut this site and it becomes a straight line, what they have found is that there's genes at the ends now, that is at the cut site, that confer some kind of protection. Okay, very edges of that circular piece of DNA, you have genes that say, hey, leave this thing alone. This piece of DNA is so Gucci in the hoochie mama that you don't want to bother it. It's like, yeah, exactly, Sparky. It's like stoppers. So Sparky, if this is correct, right, then this test works. So what the researchers did, and I thought this was super clever, you can give a bacteria plasmid. You don't have to have it use a plasmid that it naturally has. So they made a special plasmid that coded for antibiotic resistance. I'm just going to call it Ant minus, to where if the bacteria takes up the plasmid, then it will be resistant to antibacteria. Or an antibacterial. Does that make sense, Sparky? Of like, they can put that gene on the circle. If the circle is accepted by the new bacteria, then it will gain resistance. Yes, okay, awesome. Be able to make a plasmid contain DNA that makes the cell self-destruct. You could, yes. So the red part can be whatever you want it to be. You can switch whatever you want in there. There are genes called cell death proteins that in theory you could put in there and they'll get activated, but you need to have them be accepted. Okay, so if this is our antibiotic resistance segment, the really next clever experiment, Sparky, was if the caps are the protective bits, the researchers actually shifted the caps to where the cut site was now no longer at the cap point, but the cut site was a little bit outside. So let's say they cut here, right? And now those blues, actually, let me draw that a little bit better. Now the blues were all clustered at a different region, and the antibacterial resistance was up here. So the question is, which one of these will actually be protected from the cell? So Sparky, your hypothesis about them being stoppers, if those blue bits of genetic information are protecting the plasmid from being broken down, y'all tell me, what's gonna make it into the cell unharmed? Option 1 or option 2? Option 2 doesn't have the blue bits at the tips, it has it in the center. Option 1 has the blue bits protecting at the edges. So which one will be confer this protection? If their idea is right, it should be option 1 that confers the protection, because it'll be option 1 where that DNA gets protected by the blue segments. If the blue segments are not at the tips, then this piece of DNA will be broken down. Sure enough, that is what happens. This one is acquired by the cell because those blue tips, those protector bits are at the tips. It prevents the DNA from being broken down. Laura Gale with the beautiful analogy. They're like aglets for shoelaces, 100%. So Laura, if you put those aglets in the center of the shoelace, they'll get totally frayed, right? And come apart. So this segment then doesn't work. And so, y'all, this might seem really simple, and I feel like it is, but until yesterday, no one knew this. And the reason why I think it's a big deal is because we've known a lot about the immune system of a bacterium and how bacterial immunity works. CRISPR, genetic engineering, works by hijacking bacterial immunity. And yet, for 50 plus, 60, 70 years, we've been using plasmids, and no one's ever wondered why the plasmids aren't broken down inside of a cell until it was published yesterday. And I think that is really, really remarkable stuff. Now, the idea behind this and why this is such a big deal, right? Like why this is going to matter is because this study will allow the researchers to make plasmids that are more stable to be put into bacteria, right? So the genetic transfer of how efficient it would be to put a plasmid into a bacterium, we will now know that, hey, you need to add this section and it'll be far, far more stable. And then, Sparky Pugwash, you can design plasmids that genetically manipulate bacterial population by getting rid of or blocking antibiotic resistance genes. So Sparky, let's say that you have bacteria that are antibiotic resistant, and we know what genes they have that are turned on like to 11 that make them resistant. You give them this plasmid number 1, where this red section is, block gene, like a gene blocker or something, and it'll just nerf the genes that are responsible for it. Imagine the power of something like that you can fight antibiotic resistance without having to make new antibiotics. You figure out, okay, they're resistant because of this, we'll put in a blocker, and the bacteria will accept this new bit of genetic information because it's a plasmid, and it'll wipe them out. So almost like what Saphira's Mama 32 was saying, actually, right? Except it's not necessarily a cell death gene, but it's you just block the antibiotic resistance, and then that population will end up dying because then you put the antibiotics in there, and then boom, they're done. It is a very elegant and very simple methodology, and it is incredible that it was just now discovered. Something else that it could be used for is you can give a plasmid to bacteria in soil or bacteria that's in water that breaks down pollutants, or carbon dioxide fixing, or Sparky Pugwash, one of your favorite topics, the gut microbiome, right? Imagine if you could alter your gut microbiome by giving genetic information to the bacteria that might be in your gut that improves your health. And so those are all things that scientists are trying to do now, but the issue is that you just can't get the bacteria to take up the plasmid. They've now figured out, it's because you need to have this different, like this certain genetic code at the cut site to protect the plasmid. Why is it? It's like it's so remarkable that it's only being discovered now. I think it's so cool. I have some videos, y'all, that hopefully will demonstrate why this is also so cool and how some of these features work, but hopefully my explanation has made some amount of sense. Here we go. So CRISPR is what the bacteria use fight. If you just give a person a plasmid, it is doubtful that you will be able to integrate anything into the DNA itself for a human cell, but you'd be able to maybe have a circular piece of DNA that's around for a smidge. Now, one of the issues is that circular DNA is not liked by our cells. Our bodies are not designed for them. So your body would break down a circular piece of DNA because that would be recognized as foreign. But the bacteria that you'd be trying to target for the experiments that we're designing would be totally okay in being absorbing the plasmids. This video here illustrates what a bacteria normally does. So this is without that protective cap. The idea here, y'all, is that a bacteria can protect itself from foreign DNA, like if it were to come from a virus or from another bacteria. And remember, that circular piece of DNA, that plasmid, also protects itself. How does that happen? That is what we're going to be answering with this data. So, Sparky, just knowing what he's saying here, that the cell is always looking for invasive elements, right, makes you then wonder if we've known about the plasmids, like, and we've used them for, decades and decades. It's just so crazy that then no one's thought of, like, well, why isn't the bacteria destroying the plasmid? And I think that's also why this was a Nature paper, y'all, because this is a huge breakthrough in understanding fundamental biology of bacteria, but also potential biotech too. So what ends up happening, right? The bacteria integrates a piece of this foreign genetic element into its own DNA, and then that is utilized as a marker for what the cell is going to be on the patrol for. So if anything comes in again utilizing that genetic element or a copy of that genetic element, it'll be in the DNA of the bacteria, and it'll recognize it then as foreign, and then it can destroy it. So it's a means of immune memory. Remember, bacteria don't have B cells, T cells. There's no classically defined adaptive immune system, but this is a very primitive form of adaptive immunity, and it's really, really cool. Yeah, so in the experiment, y'all, we described here in our mediocre looking piece of art, option 2 would take your gene of interest and incorporate it into that immune region, and they would provide immunity against your gene of interest. So meaning that the bacterium would try to protect itself from being infected by this or have this gene present in it. And that's not ideal, right? You don't want that to happen. So this is the alternative event that'll take place. But we can avoid that by having on the edge of the plasmids those protecting bits. And so that, it turns out, that's what the bacteria do when you normally give it a plasmid. When you normally give the bacterium a plasmid, it's like, ew, I don't really want this in here. This is not something that belongs in here. Let me go ahead and nerf this. Which again is really, really cool. So Sparky, that's the really cool thing about this study, right? Is that the cells are using CRISPR, the bacteria, to get rid of the plasmids you're putting into it. And when they get rid of the plasmids that put into them, it just wipes out everything of that in that cell. That's why the plasmid efficiency and uptake is so low. But if you add in this extra component, it'll be a protector, which I think is really neat. We're talking about bacterial immunity tonight in one of the multiple use stories. So this video is going to review a little bit of the bacterial immune system and how it led to the discovery of CRISPR. Does CRISPR unzip the DNA? No, Wombat. So the unzipper is helicase. CRISPR comes in and will have a guide RNA. So it'll have an RNA sequence that matches something in the DNA. And so when the DNA is unzipped, the guide matches that sequence, and then CRISPR will attach it, and then it does a chopping behavior in there. It goes chop chop. Funny enough, y'all, this video is 8 years old, and that lady who's speaking since this video has won the Nobel Prize. Like, it's a big deal. They actually add base pairs or just an RNA strand and have it replicate those base pairs. What do you mean, Della? The CRISPR, the Cas9 molecule itself binds to a single strand of RNA, and that single strand of RNA is complementary or matches some sequence elsewhere, and that whatever it then binds to is what it ends up cutting. The DNA of the bacteria has these sequences, and they just make more copies of it as an RNA molecule. So there's base pairs free floating in the cell. It's not I'm not understanding the question, Della. I'm so sorry. How can you make an image of a protein? Computational chemistry. So Wombat, you can do what is called X-ray crystallography, and that is getting a 3D image of a protein structure, and then you can 3D model it in a computer. The structure itself in X-ray crystallography is not this pretty image that we're seeing here. It's looking at a bunch of shapes and grooves and divots and regions of a protein. You usually look at subregions in X-ray crystallography, and then you piece them together to make the full image. There's also now some computational programs that you could put the amino acids into, and it will predict the shape of the protein because of the charges, the chemical charges on the amino acids. And after you put that into the software and it builds a map of what it needs to look like, then you'll have this image. The harder thing now, Wombat, is knowing what a protein looks like when it binds to a different protein, or if it binds to a piece of DNA, because the structure is going to change. In that case, you need to have the two bound together and then doing the X-ray crystallography to see what that change looks like. You splice in the RNA to a strand, so you're not splicing in base pairs. All right, I think there's a fundamental misunderstanding, maybe. You don't splice in RNA. The Cas9 molecule holds a segment of RNA. That RNA matches to an existing strand of DNA that maybe is foreign into the cell, or it's a target of what you want it to hit. So the Cas9 is holding a single strand of RNA. That single strand of RNA matches to something it has a target. If that single strand of RNA has a target, then it can bind to that target and now make a double-stranded string of DNA. Then it gets cut. A base pair does not refer to two bases paired together. A base pair refers to an A, T, C, or G, which are the building blocks of DNA. That is called a base pair, which is what you put into anything that requires replication. Hopefully that makes sense. Yes. So an A, T, C, or a G are the building blocks of that DNA molecule. Because they form pairs is where the nomenclature comes from. They are not inherently existing as a pair in this situation. So that is a naming issue convention. The pair was that no, there's base pairs. A singular strand has base pairs. A double-stranded piece of RNA also has base pairs. It's yeah. Exactly. It's referring to the Lego fitting. Not so again, this paper takeaway from here is having the protective regions on the DNA strand of the plasmid that prevented the plasmid from being chewed up when integrated into the cell. And the idea is that this could be used for helping against drug resistance. And Della, that is also a pun because the methylation does also involve DNA wrapping. So there we go, y'all. Diverse anti-defense systems encoded in the leading edge of plasmids. And yeah, it was, again, super cool of how they chatted about it. We kind of went through the data and all the experiments that they did. We drew them out. We drew out the experiments of like how they function, what they were integrating into. A lot of the paper, actually there's this nice figure right here, kind of demonstrating their findings. So this is what we drew out, Sparky Pugwash, a little bit earlier. Here is the donor cell. Here is the recipient cell, donor, recipient. The plasmid goes from circle to linear, going from one cell to another, and then it is either protected or not. If there is leading, that protection is at the tips, then everything is totally fine. And if there is none of this, so this here is when they removed that protective cap, then there's no bacterial colonies growing here. Versus here, the caps are present and now bacterial colonies are growing all over the place. So here is the output of the experiment. Like this is the easiest way I think to see. If the plasmid is taken up by the bacteria and it gets this immune protection, or this protection from antibacterials, then you will see the bacteria grow on this dish. If it doesn't take it up, you will see no bacteria grow on the dish. So here, this has been taken up. This has not been taken up because you've removed the cap. And here they're showing like when it's like different positions of the cap, and they figured out when it's important to have more in the front than the back, and what happens if you change the positions. It for this paper. It seems really simple, and it is in terms of what the principles mean and why we care. It is surprising that it hasn't been taught or thought of until now. I think part of it might be is how much data that you needed to analyze in order to find that protective region. So it's not just that every single plasmid's protective region looks the same. The scientists identified 33,000 plasmids. 33,000 plasmids were looked at, and they found this protective cap as a common gene or gene family at the edge of the plasmid. And then when it went from one bacterium to another, as long as it had that protective coat, it would be protected from degradation in the new bacteria. If it doesn't have the protective coat, it falls apart. And so this is a means by which we can fight antibacterial resistance and maybe other types of resistance as well. So that, y'all, is your science news story number 2 on different kinds of bacterial protection and support from breakdown.